Hi, I'm Mallory.
I'm Emily.
And I'm Alexandra.
And this is our unknown 8618. This is our table of contents, and if you'd like to see it in more depth, you can check our PowerPoint. So we did some of our background knowledge on our microbe, and this is where we took most of our tests and our results. So first, this is our microbe under our microscope, and it came back as a gram-negative rod-shaped bacteria when we gram-stained it. This is our nutrient agar plate. These two samples were taken on multiple occasions, and both of them came back white, circular, and tightly packed together. And then this is our catalase test. In the video that's playing, you can see that when the catalase was poured onto our microbe, it did bubble and it did react, indicating that it was catalase positive.
Okay. So next we did an oxidase test, and this was done by putting a few drops of oxidase reagent onto our unknown, and we had a slight color change to purple, which indicated that it was positive for oxidase. And then we did a urease test, which was also positive. And so the enzyme urease hydrolyzes urea to form ammonia and carbon dioxide, and our broth had a slight color change as well in this, which indicated positive for urease. And then we did mannitol salt agar plate, and we did not get accurate results because our plate that we used was more liquidy and wasn't the right consistency, so we had no reaction. And then we did an EMB agar plate, which gave us a metallic green reaction, which was not correct, so we think we had cross-contamination. And if it had been correct, we would have gotten a pink streak on our plate, which would have meant our organism is a lactose fermentator.
So for the blood agar plate, our results were white and puffy. So our unknown appeared white and puffy on the left and right hemisphere of the blood agar plate, and this means that our microbe was a gamma hemolysis. In our slant agar plate, we had a red tip. Our slant agar plate had a red/yellow plus precipitate, which this means our microbe was a glucose fermenter plus or minus peptone catabolism, and it produced hydrogen sulfide. And then the interflurry test tube.
So this is our interflurry test tube, and this is the last test we did to kind of finalize what we had. And the code that came back for that was 70743. And so it came back positive for glucose, lysine, adonitol, lactose, and arabinose, sorbitol, and urea, and citrate, but negative for the other ones that were on the tube. And so our identification of our microbe is that we have Klebsiella pneumoniae.
Okay, so for Klebsiella pneumoniae, the modes of transmission, it's mostly from person-to-person contact, and then less commonly it's spread throughout the environment, and it's never spread through the air. And then in healthcare settings, it can be spread from contaminated hands of healthcare workers, and then also from contaminated ventilators, intravenous catheters, and wounds. And then Klebsiella pneumoniae is a common bacteria that's found in your intestines, but it's only dangerous when it's in other parts of the body. So infections are rare in people with strong immune systems, but those compromised are more susceptible.
So for infections and treatments, some infections that Klebsiella pneumoniae causes is meningitis, which is the inflammation of the membranes that surround and protect the brain. Pneumonia, which is the inflammation of lung leading to accumulation of fluid, and respiratory compromise. Cellulitis, inflammation or infection in the dermis or the subcutaneous tissues, and the urinary tract infections, which is an invasion and infection of the urethra and bladder by bacterial resident. And some treatments for Klebsiella is cephalosporins, which inhibits the cell wall synthesis, quinolas, which inhibits nucleic acid synthesis by disrupting enzymes, carbapenems, which inhibits cell wall synthesis as well, and monotherapy or combination therapy, and that uses one type of treatment or many different types of treatments, but treatments can be modified depending on which organ is involved or infected.
Okay, and then our last section is control and future prevention. So this is just going to focus on controlling or like making sure we're maintaining our Klebsiella pneumoniae, not letting it spread, and then future prevention, so what we can do to prevent it before it actually happens or gets out of control. And since Klebsiella pneumoniae is typically an opportunistic bacteria, which means when someone else is already compromised, that's when it attacks the most. Where healthy people who don't have or not like compromised already, have a better chance of not getting Klebsiella pneumoniae than those who are not healthy. Klebsiella pneumoniae is regularly found in the gut, but when it gets to other places, that's when it becomes a problem. And that's when it becomes dangerous. So, and Klebsiella is becoming increasingly resistant to drugs, which makes it even more of a dangerous, because as we're using drugs to treat it, it's getting more and more resistant, which means we have to find different drugs to treat it in order to actually control it. So then, in order to prevent Klebsiella, a big thing we do is we need to use personal protective equipment at all times. So that looks like gloves, that looks like gowns over our scrubs, that looks like constant hand washing to kind of just control and make sure that we're preventing the spread for us as nurses or us as healthcare workers, but then also us when we're dealing with other people. So like if someone is infected and then you go to the other patient, never wash your hands, never do anything, then that is going to spread it. And so we want to prevent that spread because we never know who could be like susceptible to Klebsiella, and we don't want to be spreaders of that. And then hospitals must also follow very, very strict cleaning regulations in order to decontaminate the rooms and the areas used. So you don't want to host a patient, have them in there for surgery, have them in there for treatment, whatever it could be, and then not decontaminate or not clean and sanitize the room properly, and then bring in a new patient, and then all of a sudden that person is now infected with Klebsiella pneumoniae because that first person was unknowingly carrying it. And so I think a big thing with control and prevention too is just like the unknown of like we don't know who actually has it and who doesn't all the time because may not be the reason they're in. And so just the smallest amount can infect somebody who is compromised. And so that's Klebsiella pneumoniae, and these are our resources attached on our slide. The links are in there attached into our submission document. Thank you.